There will be another lecture for our Monday holiday. I want to go ahead and do that recording. It will be made available to you before the weekend or during the weekend, sometime during the weekend. So when you come to class on Wednesday, please make sure that you review that material. And of course, there'll be extra clicker questions associated with that extra lecture. For the internet students, no extra clicker questions will occur until your extra clicker questions occur. So keep that in mind. So do be on the lookout for a Monday lecture, and it will be titled for our Monday holiday, just so that you kind of keep in track with our usual Monday, Wednesday, Friday lecture series. All right? Yes?
So the clickers will be on the video? Oh, sorry.
No. Internet students also listen to the Monday, Wednesday, Friday lectures, and so I have to make sure that when I say something about clickers for you guys, I also mention something about clickers for their guys, because suddenly I'll get a bunch of emails. Where's our clicker that you promised? There is no promised clicker until Wednesday, when you guys get clickers. So, all right. Back to the eye, and hopefully we'll be finishing up the eye soon. There is a lot of material. The eye is actually half of this chapter. You guys have done your eye eval, so all the stuff that we talked about last time is also still relatively fresh on your mind. And now we're going to go back and we're going to fill in the notes because we talked about the details. The other thing I want to make sure you guys realize, whenever you have an eval, you should be looking at your lecture notes, because lab and lecture are supposed to mesh. So whenever you have an eval that's worth points, your lecture notes will also have basically the structures you're expected to identify when you actually do that eval. Tunics, also the Latin word for coats or layers. They form the wall or the external outer aspect of the eye, and we do have 3 tunics involved with our eye. The outermost tunic is called the fibrous tunic, and it is avascular, which means no blood supply. It is dense fibrous CT. The sclera itself is irregular in its construction, but the anterior aspect of the fibrous tunic, the cornea, is actually quite regular in the construction of those fibers. So we don't have a blood supply. And in order to be living tissue, you need to have a blood supply. Where does the blood supply come from for the fibrous tunic? It comes from the choroid. So that choroid is essential to the health and integrity of that fibrous tunic. Our notes talk about 2 main regions, the sclera, which is the white of the eye, and that is essentially an extension of the dura mater. It is the toughest part of the eye, as you probably appreciated when you were trying to actually cut into the globe and dissect it out. It also covers the optic nerve. Since it's an extension of the dura mater, we'll say covers over here, optic nerve. And it protects and maintains the shape of the eye. The cornea is still a very important part of that fibrous tunic. It's, for us, one of the most important parts of the fibrous tunic. It's interesting. It's avascular, but it's obviously highly innervated. We have many pain receptors associated with the cornea, and that is actually a protective mechanism. In areas that need more protection from the outside world, we actually have a lot more sensory receptors in those areas, especially pain receptors. Your backside has less pain receptors than your front side does, and the cornea has a lot more pain receptors, so does your face, hands, feet, things along that line. Structure of the cornea. It says convex, modified, transparent part of the fibrous tunic. So transparent means light can go through it, and indeed the cornea is that structure which bends light the most. So we'll get into refraction, because that's what refraction is all about. It's all about the bending of light. It is interesting. Cornea is not covered with conjunctiva, correct? But it does have a protective covering. And it's the same type of protective covering that you experience with your skin. It's stratified squamous epithelium. So stratified squamous epithelium, as you know, is designed to be sloughed, and indeed this outer aspect of the cornea is designed to do that. So that's our outer protective layer. And also remember with the skin, that's where we actually have the epithelium is innervated, and so we have a lot of pain receptors in that area. This stratified squamous epithelia joins with the conjunctiva at the scleral corneal junction. This is what is traumatized. That's why it's there. Traumatized when somebody gets corneal abrasions. Say experience. Come on, speller. Okay, so when somebody gets corneal abrasions, it hurts like heck, and your tears have what in them? Salt. So that hurts too. So the thing you want to do is you want to just close your eyes. So closing your eyes is actually applying your own homemade Band-Aid to it. As long as you keep it covered, it will re-epithelialize for you, and then you'll be just fit as a fiddle after that re-epithelialization actually occurs. So stratified squamous is the source that protects the cornea. Conjunctiva is the source that protects our sclera and keeps things from actually entering into the orbital cavity. Yes?
How long would it take to heal an abrasion?
Corneal abrasions have a very high, the stratified squamous epithelium has a very high regenerative rate. And depending upon the degree of corneal abrasions, 24 and sometimes occasionally 48 hours is all you need. So when people get bad corneal abrasions, they go to the eye doctor because that hurts like heck, and you want some numbing medicine is actually what you want. And they slap a Band-Aid, they close your eye and slap a Band-Aid on it, you know, an eye guard on it, and tell you come back and see me in 24 hours. Because you know by that time you'll actually be pretty much all repaired. On the inner aspect of the cornea, what's really fascinating, it's just a single layer of simple squamous epithelium. That's kind of like our blood vessels. You know, simple squamous epithelium. What's interesting about this simple squamous epithelium, these cells have sodium pumps in them. And these sodium pumps are designed to actually keep the sodium content of the cornea low. When we keep the sodium content of the cornea low, well, let's talk about if these pumps weren't working. So that means that sodium can actually infiltrate the cornea itself. When sodium infiltrates the cornea, what also follows sodium that we talked about all the time? Water. So what happens to the thickness of the cornea then? Will that cause it to change? So the cornea is a specific thickness, and it's always that thickness. Okay. That stratified squamous epithelium is just a protective layer. So these sodium pumps on the inside, lining the inside, are designed to make sure that the sodium concentration of the cornea remains low so that water doesn't get into that. Now, this is also, because if water got into the thickness of your cornea, what would happen to your vision, your visual acuity? It would definitely change. It would become much blurrier. It's like somebody put a pair of glasses on you for correction that you don't need. So this is an interesting phenomenon that you have to remember. When we talk about movement of water, it's because of solutes, right? That osmolarity kind of thing. Wherever the solutes are in higher concentration, that's where water goes, and water can cause swelling and things like that. Remember this red blood cells popping or, you know, shrinking up? Well, with these sodium pumps, they're just specific for sodium. So there's a condition when people get type 2 diabetes. Type 2 diabetes means what's elevated? Glucose. Glucose is elevated. Where is it elevated? In the bloodstream only? No. You get sugar-coated everywhere, okay? Glucose is everywhere because glucose has the ability to move everywhere, right? So glucose is everywhere. And so glucose is high in the aqueous humor, which means that it can actually penetrate your cornea. If glucose penetrates your cornea, what can also then go with it? Water, because glucose itself is a solute, correct? Some sort of particle, it's something that's dissolved into the liquid. And so when people have type 2 diabetes and they have yet to be diagnosed and properly treated, guess what they start to complain about besides being very thirsty and peeing a lot? They have blurry vision. So the doctor tells them, I need to go see the eye doctor. The doctor says, don't go see the eye doctor yet. Let's wait until we get your glucose under control, and then the fluid in the cornea will actually start to decline, and you'll have your visual acuity actually restored. So these types of things you guys have started to learn about. You just haven't learned how to actually take them to other areas that and apply them. So it's important. Now, whenever the eye is then plucked out of the head, it's obviously then dead. So what do you think happens to the activity of these sodium pumps? Whoop, they go away, right? So the sodium that's in our aqueous humor then gets to penetrate into the cornea, and water penetrates into the cornea. And what do you guys notice in those corneas of animals that have been dead for a while? They start to get cloudy. And that cloudiness is actually a part of this phenomenon. There's more to it, but this is part of the phenomenon. The cornea is also just like the sclera, because it says our fibrous tunic is avascular. It's been reiterated, it is avascular. That also tells you that it's not accessible to the immune system. And this is really important because of this nature, avascular, and the immune system has never really actually encountered or met it. You can transplant freely. Without rejection. So people who get corneal transplants don't actually have to be on lifelong anti-transplant medication. So this is what's really cool. And so when people pass away, it's not uncommon for them to actually donate their corneas. And so the team comes in, obviously not long after death, otherwise they'll start to have problems with the corneas. They'll come right on in and they'll go ahead and they'll shave those corneas off of them. And then because they shaved the corneas off of them, you've actually kind of exposed the inner aspect of the globe. And so the eyes want to depress. And so they put little round, kind of like wiffle ball covers over that area so that the eyes remain relatively protruding instead of sunken. Because everybody doesn't want to look at, if they have a casket, open casket viewing, nobody wants to look at their loved one with sunken eyeballs. So that's what the transplant team comes in and does. So whenever we get our cadaver, we never know if they've had a corneal transplant, if they've been a donor for that, until we actually lift up those eyelids and take a look, see whether or not they have that little wiffle ball layer over that area. So it's very nice. They take those corneas, you can go ahead and treat them almost like a lens. You just freeze them, you grind them down to the size and thickness that you need, and then go to the person that you're going to transplant it onto and roughen up the surface and put them on like contacts, and then they actually grow together nicely. It's fascinating. Eyeballs are fascinating, aren't they? Well, 70% of our sensory information of the world is coming to us from our eyeballs, so there's a lot to them in that relatively simple structure. The next layer is called the vascular tunic. It's also referred to the uvea, our middle coat of the eye. It has three main subdivisions or regions. The posterior segment or posterior cavity has the choroid. Then as we move anteriorly, we'll come across the ciliary body, and then as you move in front of that, you'll next find the iris. So all of those three structures are part of that vascular tunic. Now, the choroid is, let's put in our notes, vascular part of posterior cavity. feeds the fibrous tunic, which you guys correctly identified. There is a lot of dark brown pigment in our choroid, and this is important because it's designed to prevent light scatter. When you take a look into somebody's eye, a human's eye, it doesn't have the tapetum lucidum like our animals do, that we actually see the reflection of that tapetum lucidum in low light situations when light actually strikes it. Those animals are actually foraging, feeding, or trying to avoid being fed upon. And those animals will have that tapetum lucidum to help them actually maneuver in low light situations. And so the tapetum lucidum is designed for light scatter. The thing about that is that light scatter in their eyes helps them with their world around them, but does not provide them the clarity that we experience without the tapetum lucidum. We are able to actually get very little light scatter and get those light rays actually nicely focused on our retina to give us the information that we want in a precise fashion. Those animals with the tapetum lucidum don't have beautiful crisp surfaces like we do. Their surfaces have more blurriness to them. We have lovely crisp edges to whatever we're looking at in good light situations. In low light situations, because we don't have the tapetum lucidum, we have difficulty seeing what other animals obviously see in the low light situations. Let's go to the ciliary body. We're moving forward now. Continuous with the choroid anteriorly. Lots of dark brown pigment in that too, wasn't there? So that dark brown pigment, it goes all the way up to that, but it's not going to have a major impact on light scatter. It is mostly smooth muscle, and its processes secrete the aqueous humor. Now, the aqueous humor is secreted in which chamber? Posterior chamber. Goes through the pupil into the anterior chamber, and aqueous humor, remember, is produced 24/7, and so the overflow valve is located at the sclerocorneal junction in the sclerocorneal in the scleral venous sinus. An old-fashioned term for it was based upon the person who identified it, and Schlemm was the guy that identified it. It was called the Canal of Schlemm. I actually like the Canal of Schlemm. It's kind of fun to say for some darn reason. Rolls off the tongue pretty well. But it's the scleral venous sinus, sometimes referred to as the Canal of Schlemm. That's for the overflow of the aqueous humor. Off of the ciliary body are these suspensory ligaments coming off of the ciliary processes. They suspend the lens in the middle, right in front of that donut hole, right in front of your pupil, and are responsible for providing tension on the lens. Now, the lens is in a nice fibrous capsule. It's actually a fibroelastic connective tissue capsule, which actually makes sense since it's so mushy, squishy, and gelatinous. And so these suspensory ligaments are on the perimeter or periphery of the lens, on the periphery of that capsule. All organs always have a capsule associated with them, and so the lens is actually not unusual in that aspect itself. And the tension on those suspensory ligaments will determine whether or not the lens is flat or if the lens is fat. Flat or fat. So what's the tension like on the suspensory ligaments if the lens is fat? Low. And if the lens is flat, high. So that's the thing to always remember. The lens is what helps us with basically our near vision. It's designed to actually have that flexibility associated with it. And so when we want to take a look at something up close, just intuitively, I want to see if you guys intuitively know, which do you want? If you want to look at detail up close, do you want to grab a skinny lens or a fat lens? Fat lens. And that's what our lens does. Whenever we want to look at things up close, our lens gets fat. And so we're able to actually see the nice detail. And so our lens overall, for the most part, is supposed to be flat, because flat helps us to actually survey our world to make sure that nothing dangerous is coming after us. And so in our society, though, we depend upon fat lenses, don't we? Because we do a lot of things up close with our hands. And so we have a lot more conditions related to that process, where we end up losing some of the flexibility of our lens because we make it fat too much. Yes?
Does squinting release that tension?
Squinting has nothing to do with it. Squinting will only enhance the process that we're going to talk about, I don't know if we'll get to today, called accommodation. And accommodation is actually the fattening of the lens. And one of the things I will tell you now, since we're talking about it, is that do you guys remember as you went higher and higher on power on the microscopes, whether your field of view got bigger or got smaller? Got smaller. Guess what your pupil does? Gets smaller. The closer you get, the smaller your pupil gets. So it's very fascinating that our microscopes in our eyes have a lot of similarities there. So you caught me when I was actually recently thinking about that. The iris, still part of our choroid or our vascular tunic or uvea. It is the visible colored portion of the eye. When we say what color of your eyes, it's shaped like a donut because it has a hole in the middle that we call the pupil. It is the color of the iris is related to the amount of dark brown pigment within it. Infants, Caucasian infants, do not have much of this nice dark brown pigment, so their eyes are almost always blue. So it's the interaction of the blood vessels, the vascularity, the muscles, as well as a little bit of dark brown pigment that gives newborns their blue eyes. As you age, whatever your genetic destiny is to be, based upon the genes that your parents gave you, you will get more and more dark brown pigment depositing into the iris, and therefore your iris will change its color. What do you suppose happens as you age? Eyes start to actually get a little bit paler in their color because you actually start to lose some of that pigment. So you can, every 10 years, you should check your spouse out and take a look at their eyes. So when they ask you what color are my eyes, they know you're paying attention to. If you're an albino, what color is your iris? It's pink because you have no pigment whatsoever. So that's why you have the blue, because you do have some. But as you age, typically about age 2 or 3, you'll have the color pretty well established of what you're going to be, whether you're going to be blue-eyed child, or hazel or green, or brown eyes of various shades or hues. So that's the natural course of things. Again, pupil is the center. And the size is controlled by the iris. This is smooth muscle. Surprise, surprise. The ciliary body was smooth muscle, wasn't it? This also is smooth muscle because it is not under our voluntary control. And one of the things that is different about this smooth muscle is its control mechanism. This smooth muscle acts like what kind of muscle in your body? Skeletal muscle. So you have an immediate response. So do you remember multi-unit versus unitary or single unit? Which one gives you the peristaltic wave? Is it multi-unit or unitary or single unit? Single unit gives you the peristaltic wave. And so our iris, the innervation to our iris, is not the single unit or unitary wave. It is actually the multi-unit innervation. We have the smooth muscle fibers going in two distinct ways. We have radial ones, and we have circular ones. And let's stop for a moment and think about when the circular ones contract, what happens to the size of the pupil? Not even looking at the picture, we're doing conceptual thinking. What happens to the size of the pupil when the circular ones contract? Gets smaller, okay? And so when we want to actually dilate or open up that pupil, make it bigger, the circular ones obviously must relax, and then which ones are going to contract? The radial ones. Now, to remember when you're in fight or flight mode, because that's what sympathetic innervation is all about, right? Sympathetic nervous system's fight, flight. Parasympathetic nervous system is rest, digest. Do you guys still remember that from last semester? Okay. What do you want to do? If you're running away from the bear, do you want to have a small pupil or do you want to have a big pupil giving you as much information about your world as possible? Big pupil. So if you, that's why I try to get you guys to do more than just strictly memorize. If you do conceptual storytelling to yourself, it's so much easier to actually remember what it is. So our radial fibers are under sympathetic control and our circular fibers are under parasympathetic control. Rest, digest. You're eating your food. You don't have to worry about running away from it, so your little pupils can get little. You know, but suddenly something's coming at you to attack you. You want to see where to go. You're going to open up that lens and you're going to, or that area, not the lens, but you're going to open up the pupil and be able to get more information from your outside world. So radial sympathetic, circular parasympathetic. So when somebody comes to you and they've got dilated pupils, you go, whoa, for many different reasons, right? Yes.
So what's happening when you have tunnel vision?
When you have tunnel vision, well, that's because that's circulation to your brain is actually being restricted. You're getting ready to pass out and faint when you get tunnel vision. That's actually a occipital cortex issue. Yeah. Anybody ever faint in here or willing to admit it? Yeah. Did you notice that the world was the outside of the world was fading away? Do you remember that? Yeah. Most people that are actually having what I call a slow faint, and they're gone. Lights are out. Retina. That's the innermost tunic. It also has two distinct layers. It has an outer pigmented layer, which is right adjacent to the choroid, so surprise, surprise, pigmented layer next to another pigmented layer. And it is also designed to help to absorb extraneous light. We do not want light scatter. We do not want light scatter. The other important thing about that pigmented layer, it is the layer that produces vitamin A. And vitamin A is absolutely essential for healthy photoreceptors, healthy rods and cones. So that's why your parents tell you, eat your vegetables or your fruit, because vitamin A comes from what's the fruit source or vegetable source that we need to ingest in order to have adequate amounts in our diet. Carrots. It's those colorful fruits and vegetables, right? So anything that's red, yellow, orange, that kind of stuff is going to have that carotene pigment, which your body will then take in and produce vitamin A. And then vitamin A is an important component of healthy rods and cones. If you don't have adequate vitamin A, then what does that mean? You'll have unhealthy rods and cones, and in the world, there is a problem sometimes getting colorful food, right? There are some places that have difficulty obtaining a well-balanced diet. Yes, that's still present in the world. And so those countries that have difficulty getting a well-balanced diet to all their peoples actually have a high degree of blindness because of this. They're not getting enough precursors to make vitamin A to help feed their rods and cones and make them healthy. And so sometimes you'll see stuff, because I know some people are very, very anti-GMO, and I look at my dog and she's a poodle and I go, "You're a beautiful GMO dog. I'm glad you're here." You know, but you can genetically modify a breed by many decades of crossbreeding, or you can do it in a lab. You know, you're doing it one or the other way. So there are labs that are working on creating a rice called yellow rice. Guess what's in yellow rice? The carotene, the precursor for vitamin A. And so if you put into the plant the ability to actually in itself give you your precursor to vitamin A, you actually then can distribute this crop to impoverished areas that have rice as a staple to their diet, correct? So sometimes when you see these things about GMOs, read a little bit more about it before you go anti-GMO stuff, because it's better to give people stuff which will give them vision than to let them go blind. Any questions? No? Okay. That's my soapbox for GMOs. All right. Neural layer. It too is transparent. When you guys took a look at it in the lab, it didn't look transparent at all to you guys, right? It was opaque, but that's because we are no longer living and it's also preserved. So interestingly, it too is transparent. It's a nervous layer and it's an out pocket of the brain. And so I liken it to this. The brain was a glob, you know, in its formation, and it grew out these two little feelers, and these two little feelers hit this area that started to actually form the lovely fibrous and vascular tunic around it, and then it went boom, and it lined the inside of it. And so you see with the retina, when it's collapsed, you're basically looking at almost a form of the optic nerve. So it's the absolute extension of that optic nerve itself. As far as the cells of the retina, we have photoreceptors adjacent to the pigmented layer. The photoreceptors are either rods or they're cones, and we have 125 million per eye, so 250 million total. The photoreceptors are then connected to bipolar cells as we're going towards the center of the eyeball itself. And then bipolar cells then synapse or connect to our ganglion cells. And then they have axons going over the inner surface, and remember the inner surface is adjacent to the aqueous, the vitreous humor, to form the optic nerve. So this is our cartoon model right here. Let's look at the histology that you will be putting in your histology atlas if you have not already done so. So we see in our notes here choroid, and it's easy to figure out what the choroid is just by looking at that picture, correct? You see pigment and you go, that's got to be choroid or that pigmented layer of the retina. Which one is it? Well, it's choroid because the pigmented layer is really, really thin. You see that? It's just that line right there. Then we see the nuclei of our rods and cones. We have it on the other side, but you know what? It just helps when you write things down. So it doesn't hurt you to write it on down. So those are the nuclei of the rods and cones. They have their tips buried in the pigmented layer. So the stuff where light is going to hit, is right in here. Light hits that. But light has to go through the nuclear layer of the rods and cones. And then light also has to go through our bipolar nuclei. So here's where our bipolar cells are. And then here's the ganglion cell layers. And the ganglion are actually having their axons leave and go towards the optic disc. So all the axons are going off in this direction. So they are also on the surface of the retina. So what I like about this picture over here is that, let me make sure I do it like that, that it shows you light is coming in and going through all those layers to stimulate the receptive part of our photoreceptor cells. Then the information going to the brain is in the opposite direction. So light has to actually pass through quite a few cellular layers before it actually hits our photoreceptor cells. And so we want to have precision in actually how that light gets there for us. The optic disc is indeed the area where the optic nerve is exiting the eye. It is a collection of axons from the ganglion cells. Converge here. It's the only area where the retina is actually attached, so to speak, correct? Because the retina is not attached to any area except for this one area. And that's because that's where everything is actually going out. And no photoreceptors here. And because of that, this is referred to as the blind spot. Are you aware of your blind spot ordinarily? No. And you have two blind spots, correct? Okay. So let me ask you a question. Where are your blind spots really located in real time, real anatomy? Where are your blind spots really located? Are they located off to the side in real time in your eyeball, or are they located straight back, or are they located nasally? They're located where the optic nerve is, right? So where's your optic nerve? Remember the optic chiasm? It's nasally, right? So your blind spots are medially or nasally located. Your macula lutea is straight back. Your blind spot's nasally. Where do you experience your blind spot? Laterally. You're like dishes, you know, those dishes for satellite TVs and stuff like that. Your retina's lining this cup, okay? And so if something's off to the right, it's going to strike the retina where? The lateral aspect if it's the left, correct? The medial aspect if it's the right. So whenever you are using both your eyes, are you not getting information still from the other eye where the optic disc is not giving you information? Do you guys see this? So you have two eyes, you have two optic discs, you have two blind spots. They're both nasally located, and the only time that you actually have problems with them is if the other eye is not able to give you that information to fill in what the other one is missing. So that's why you have car accidents, correct? You have a car accident because I didn't see you, because they were in your blind spot, which means only one eye was capable of seeing that area where they were in. You guys follow me with that? So that's why you're unaware of your blind spot, because you have two eyeballs. Your blind spots are lateral in one eye and lateral in the other eye, but the information is coming to you for whenever something's in one eye's blind spot, it's not in the other eye's blind spot, as long as you can still see that object. So you always have that information filled in. Now, the next thing, the macula lutea and fovea centralis, lateral to your blind spot. Well, where did I say your blind spot was located? Nasally or medially. And your macula lutea and fovea centralis are like those guys on the airplane tarmac. They're saying, straight, keep going, straight ahead. That's where your macula lutea and fovea centralis is. Macula is Latin for spot. Lutea is yellow. And so when you take a look in somebody's eye, a live person's eye, and are examining their retina, the macula lutea, you can tell what it is because it has a yellowish hue compared to the rest of the nice orangish, pinkish red retina as you're taking a look at it. So small oval spot, yellow, straight back. So if you're looking straight ahead, the macula lutea is getting activated by the information coming in. But what about off to the side? Is it macula lutea being stimulated by stuff coming off to the side? No, because that's going in different directions, angular directions through the pupil and hitting that lens. That's why we are hitting the lens and then hitting the retina. That's why we want to have certain bend to our light rays. Now, what's interesting in the macula lutea, that is an area that is mostly cones. So mostly, does that mean all cones? No, because the only area that is only cones is in the very center of the macula lutea called the fovea centralis. What's interesting about that fovea centralis is cones are there and with no bipolars or ganglion cells. It's a very small area. You have to take a microscope to actually see it and identify it. You can't tell when you're taking a look at somebody's retina in a living individual. You actually have to have special equipment to actually see it. So it's an area where light hits the cones immediately. So it is going to give you the best clarity. The fovea centralis is the sweet spot. It's like the bullseye target, and the one right in the middle, that's what you want to get, the fovea centralis. It is the area of best visual clarity. Looks like dairy. Now let's go back to the macula lutea. Mostly cones, and when we get to from the macula, cones gradually decrease until we get to the edge of the retina. So when I draw my picture of this, my cartoon, here's my macula lutea. That little guy there is my fovea centralis. So mostly cones. Does that mean there's no rods there? No, that means there's a rod or two or three, but it's mostly cones. Then I get out here and I'll say still more cones than backwards. Wow. I'm on a roll today if I can do that. Then rods. It's pretty bad, but you guys know what I'm trying to do. I always hope that you know what I'm trying to do. Oh, see what happens when I try to erase? Yeah, make it better. Mostly rods. Or plus some, plus Some rods. Okay. And as we go further out, we get more rods, right? More rods than cones, less cones. So when we get to the perimeter of our retina, that's made out of all rods. Now, what defines the end of the retina? Because is the retina completely filling up the entire inner aspect of the orbit? No, the globe, I mean. So where does it? Ora serrata, excellent. The beginning of the ciliary body also marks the end of the retina itself. Now, the other thing we'll talk about is internal cavities and fluids. And you guys already know this. Posterior cavity is that area behind the lens filled with the vitreous humor. Vitreous humor is also called the vitreous body. It's indeed a clear, transparent gel within that posterior segment or cavity. And I put segment here because in the old notes and old exams, segment was the word that we used instead of cavity, which we're using right now. The vitreous humor does support the eye shape. What you're born with is pretty much what you have for the rest of your life. It is clear, so it transmits light. So light rays easily pass through it. It is there also to keep the retina in place. And it supports the lens posteriorly. Did you guys notice how the lens kind of stuck to it? Anterior cavity divided into two chambers. The anterior chamber in front of the iris, so the iris is the dividing line between our chambers. Posterior chamber is behind the iris, but in front of the lens. The fluid that we find there is the aqueous humor produced in the posterior cavity. That's where the ciliary processes are. It's produced 24/7. PD for produced 24/7. Goes out through the pupil into the anterior chamber and out of that area via the scleral venous sinus or the canal of Schlemm. If you don't get drainage, what will happen? You lose vision. That's called, you guys know? It's called glaucoma. So when you hear the term glaucoma, what they're telling you is a person is getting visual loss because they're not getting adequate drainage of the aqueous humor from the globe, from the eyeball itself. And why would that cause you to lose vision? Pressure. It's like an elephant sitting on wet toilet paper. It's destroyed. And that's where our neurons are to give us the information and also the photoreceptors for the information for vision itself. So we'll go ahead and I'll stop there and create another YouTube for the Monday lecture.